Welcome to the Apron Academy. This video is specially designed for the dietitian in training, also known as the RD-to-be. In this video, we're talking about zinc. So after potassium and magnesium, which you can watch those videos separately, but after those, zinc is the most common intracellular metal ion. So where can we find zinc? We can find it in red meats, organ meats, so things like liver, shellfish, nuts, and legumes. So fruits and vegetables are typically low in zinc. So zinc only has one valence state, so it looks like this. That is the one valence state of zinc. It serves mostly structural roles, so since it doesn't have different valence states, it's not like zinc 2+, zinc 3+, it can't really function in redox reactions. So primarily structural roles, and we'll see that as we go along. So zinc is absorbed in the small intestine and primarily in the jejunum. So there can be two ways that it gets absorbed. If we have a high concentration of zinc, it can diffuse passively, or this paracellular diffusion. So it goes right in between our enterocytes. But also if we have a lower concentration, it can go through transporters into the enterocyte. So ZIPs, this ZIP4, can bring it into the cell. This zinc transporter 5B can also bring it into the cell, but typically the transporters can also bring it outside of the cell. So you see that arrow going that way. Same here, the transporter can take it out of the cell, and zips bring it in. So here I have ZIP4. ZIP4 seems to be the main transport mechanism because we can see if we have a mutation in ZIP4, that can lead to acrodermatitis enteropathica, or AE. So if we don't have any ZIP4, we have problems absorbing zinc into our intestines, which leads to AE, dermatitis, poor growth, alopecia, and immune deficiencies. So we need our ZIP4s. And then once it gets transported into the blood, it actually binds to albumin, and that's how it travels throughout the rest of the body. So at a high zinc status, excess zinc absorption is inhibited through the production of metallothinning. So we'll talk about that a little later. But that is a zinc storage protein. So we see here in the cell, zinc is binding to metallothinning, and that helps us storage. So metallothinning is found in the cytosol and then produced due to high zinc intake. In the enterocytes, metallothinning acts as a block, preventing movement through the cell and limiting the absorption as cells are sloughed off. So we've mentioned before with iron that if we have storage in our enterocytes and it doesn't leave the cell, then enterocytes have a life of about 3 to 5 days. So if our metallothinning is keeping our zinc inside the enterocyte, eventually our zinc will be excreted because our cells are dying off and getting rid of each other. So with high zinc, we inhibit intestinal copper because copper has a higher affinity to bind to metallothinning. So if we have high zinc, it's going to mess with our amounts of copper. So high zinc will stimulate the production of metallothinning, reduce copper absorption, And then if we have high copper, it doesn't go the other way. So where high zinc reduces copper absorption, high copper does not reduce zinc absorption because zinc is a stronger inducer of intestinal metallothinning. So once it gets in the body, how is it absorbed? So copper and zinc are bound to albumin, like I mentioned before, once they're in the blood, and then they're transported to the liver. And so after reaching the liver, zinc is repackaged and released into circulation and bound to alpha-2 macroglobulin. So with distribution, about 75% of zinc is bound to albumin, 40% is bound to alpha-2 macroglobulin, and 3% is our low molecular weight ligands. So 24 mammalian zinc transporters have been identified, and they don't even require energy for function. So that just shows the importance of getting zinc everywhere in our body. These transporters don't even require energy in order to function. So after endocytosis, when albumin is taken up, zinc is released across endosomal membrane into the cytosol, brought in by zips. So you see all of these are zips. It's brought into the cell. So in the cytosol, zinc can bind to zinc-binding proteins, including metallothinning, and zinc can leave the cytosol by zinc transporters. So this here is a zinc transporters, and it can leave by this. So zips can take zinc into the cytosol, and zinc transporters take it out of the cytosol. And here we have, this is kind of showing that zinc can enter even when it's bound to albumin through endocytosis. So it can come in through endocytosis bound to albumin or through these zips. All right, so what does it do when it gets there? Kind of talking about storage. So we mentioned just a minute ago that this metallothinning acts as storage. So now we're going to talk about that in a little bit more detail. So if our diet is deficient in zinc, our status rapidly declines because we don't really have any big storage pools for zinc. So this is where metallothinning comes in. So metallothinning acts as a zinc storage protein, and under normal conditions, zinc is primarily the primary thing bound to metallothinning, and no metal-free metallothinning exists. So metallothinning, which is like this little bow tie looking thing, rapidly is induced with high levels of zinc or heavy metals like cadmium. So here I have my little circles. Those are my little zincs, and these squares are the cadmium. So binding to these metals reduces their intracellular concentration. So they're stored here, they're not going out into the cell. Hepatic metallothinning is induced by glucocorticoids and Li6, causing a redistribution of zinc from the plasma to the liver following a bacterial infection, rheumatoid arthritis, or intense exercise. So zinc promotes metallothinning gene transcriptions through metal response element binding transcription factors, that is MTF1, and MTF1 drives the expression of metallothinning. So with low zinc conditions, metallothinning is bound to this zinc-sensitive inhibitor, and when zinc is reintroduced or we have higher zinc levels, we have this free zinc. And free zinc is not bound to metallothinning, which causes a dislocation of this zinc-sensitive inhibitor, and then MFT1 is released from that inhibitor. So since that's released, this causes the MTF1 to be able to interact with this metal response element in the metallothinning promoter sequence. So that can help activate the metallothinning gene and promote that gene expression. So transcription of metallothinning is also induced by cadmium and hydrogen peroxide. So this can help when we have free radicals. Hydrogen peroxide is produced, so then we induce metallothinning, which can help kind of grab any of that. So, still want to be here. So, copper and cadmium binds to metallothinning at a higher affinity than zinc. So, if we have higher levels of cadmium, it has that higher affinity to metallothinning, and so zinc is kind of pushed out. So, whether we have high zinc levels, or even if we have high cadmium levels or high copper levels, then it's going to choose to be within the metallothinning, pushing zinc out. So then the zinc can be displaced. We have the free zinc, which binds to that MRI, or that inhibitor, that zinc inhibitor. Okay, so I just want to go over this just one more time just to make sure that it's very clear. So in a normal cell, zinc is in our metallothinning. If we have increased zinc, or like we talked about, increased cadmium or copper, it pushes out free zinc. And in the cytosol, now separately, in the cytosol, MTF1 is bound to a zinc inhibitor. So we call this an MTI. And we have this MTI inhibitor, and free zinc binds to that inhibitor, which causes it to break free from this MTF1. MTF1 then moves to the metal response element and drives the expression of metallothinning. So this is how high zinc increases the expression of metallothinning. So now, just briefly, I want to talk about the excretion. Zinc can't be put into bile. We actually have pancreatic enzymes that are responsible for digestion. They're in the lumen of the intestines, and zinc is there because the pancreatic enzymes require zinc for function. So since they're in the lumen, we're actually able to reabsorb zinc. Okay, so now I'm going to talk about one of the functions of zinc, which is zinc finger proteins as gene transcription factors. So zinc gives transcription factors this very specific structure. So we talked about how one of the functions of zinc is a structural function. And so these gene transcription factors have these finger-like structures. So these zinc finger structural molecules are able to bind to response elements on our DNA, and 10% of the human proteome is made up of zinc binding proteins. So and 27% of those are the zinc finger proteins. So these zinc binding to these zinc fingers permits the folded region to bind to the DNA sequences in the promoter region. So these fingers do not bind to the DNA in the presence or in the absence of zinc. So we need zinc in order to do this. So again, zinc causes the folding of DNA, so zinc fingers can bind, and they are always able to fit into these grooves of DNA to alter gene expression. So this is why we have so many zinc transporters. In order to get zinc, in order to help with gene expression. So what happens if I don't have enough? I like this picture. You can look at it. It talks about zinc excess, but also zinc deficiency. So the primary feature of zinc deficiency is slow growth, failure to thrive, teratogenic effects, and skeletal effects. So 10% of our DNA is regulated by zinc. So that means it's very important to have our zinc. Another way that I guess zinc functions, and so it's very important that we're not deficient in it, is through immunoregulation by zinc and also copper. We'll talk about this in the copper video. So it's important for the differentiation of various immune cells. So zinc and copper impair immune function due to loss of lymphocytes, which is a result of zinc, and neutrophils, which is needed with copper. So our monocytes also require zinc for the secretion of pro-inflammatory cytokines. So if we don't have the correct ratio of immune cells, like enough lymphocytes or monocytes, then it might mean that we are deficient in zinc or copper. So there's not really a reliable assessment for zinc concentrations. If you look at labs, it kind of looks normal, except under severe conditions of extreme deficiency. In those cases, we may see acute inflammation, serum zinc levels fall, or under acute inflammation, we see zinc levels fall, and then copper and ceruloplasmin actually increase. So we could check our hair. That's more for long term if we have a zinc deficiency or toxicity, but really even nowadays hair color and hair products can affect how our hair displays some of those things. So what a deficiency could look like is a loss of appetite, loss of normal taste sensation, alopecia, hyperkeratinization, and reproductive abnormalities. With a toxicity, it could look like having a metallic taste, headache, nausea, vomiting, epigastric pain, abdominal cramps, and bloody diarrhea. So also with toxicity, we have a decreased immune function and altered copper and iron status. So with an ingestion in high amounts, we can also see an inverse in copper deficiency. So when zinc goes up, copper goes down, which can also cause neural problems. So as you can see, zinc is super important for our life. We need it for all of that gene expression and for our DNA. And it's important to have the right amount. So if you found this video helpful, please like, subscribe, and share this video with others. Thanks.